Dr.Drillon from Memorial Sloan Kettering Cancer Center, and on behalf of my colleagues, I'm proud to distill for you the take-home messages from our manuscript, SHP2 Inhibition Sensitizes Diverse Oncogene-Addicted Solid Tumors to Retreatment with Targeted Therapy, published in Cancer Discovery. PF07284892 is a highly selective, allosteric, potent SHP2 inhibitor with a fused dihydroindene phenyl that fits deeper into SHP2, forming enhanced contacts with several residues as shown on the left. Preclinically, efficacy is maintained despite intermittent dosing every 2 to 3 days as shown in the middle. And finally, on the right, you'll see that brain penetration is improved compared to other SHP2 inhibitors. In 3 oncogene-driven cell lines with an ALK fusion, BRAF V600E mutation, and KRAS G12C mutation, treatment in vitro with PF07284892 in combination with oncogene-matched targeted therapy led to maximum inhibition of phospho-ERK levels 60 to greater than 90%, compared to PF07284892 alone or matched targeted therapy also when given alone. Consistently, as shown here in this slide, maximum tumor inhibition was achieved in corresponding mouse xenografts only when the SHP2 inhibitor was combined with oncogene-matched targeted therapy. Recognizing that SHP2 inhibitor monotherapy was unlikely to work well based on this preclinical data, a phase 1 trial was mounted with a design that distinguished itself from many other typical phase 1 trials. Specifically, unlike traditional dose escalations that would have first explored monotherapy alone and only later moved into combinations as shown on the lower left, this trial allowed the early introduction of combination therapy from the get-go in dose escalation, as shown on the upper left and the expanded panel on the right after 2 cycles of SHP2 inhibitor monotherapy. The targeted therapies added to the SHP2 inhibitor were lorlatinib for ALK or ROS1 fusions, cetuximab and encorafenib for BRAF V600E mutations, and binimetinib for MAP kinase pathway alterations. Importantly, this forward-looking design certainly paid off. At the very first dose level of the trial, 4 patients who unsurprisingly progressed on SHP2 inhibitor monotherapy were treated with combination therapy that rescued disease progression and achieved a partial response. These heavily pretreated tumors were an ALK fusion-positive lung cancer, a ROS1 fusion-positive pancreatic cancer, a KRAS mutant ovarian cancer, and a BRAF V600E mutant colorectal cancer that had progressed on targeted therapy administered prior to the trial. On many other studies, these patients would have only been given monotherapy during dose escalation and sadly come off trial for progressive disease. In conclusion, PF07284892 is a novel, potent, selective, and brain-penetrant SHP2 inhibitor whose preclinical activity is maximized in oncogene-driven cancers in combination with match-targeted therapy. The drug's phase 1 trial was designed in a non-traditional fashion to allow early combination therapy that rescued monotherapy progression in several patients with a variety of oncogene-driven cancers. This study design serves as a model for future phase 1 studies of novel known combinations, particularly when one agent is likely to be ineffective as monotherapy. And this paper in general highlights the power of strong collaborations between multiple stakeholders, including academic and industry partners. Thanks for your attention.